right, welcome back to part 3 of this presentation. So now that you have had some practice using the vocab and doing some Punnett squares, we are going to be talking about other types of inheritance patterns besides complete dominance. So remember, complete dominance is when the dominant allele is fully dominant over the recessive allele. So if you are heterozygous, you will show the dominant phenotype. For this dominant pattern, for this inheritance pattern, this is called incomplete dominance. So what this means is that the alleles or the trait will blend together if you are heterozygous. So the easiest way to explain this is with like flower color. So if you cross a white flower with a red flower, the offspring, the traits will blend together and you will have a pink flower. So that's the alleles blending together because red and white makes pink. So let's do this in an actual Punnett square so that we can better understand how this works. So like we said, flower color can follow an incomplete dominance pattern. So if we take a red flower, which its genotype will be big R, and a white flower with the genotype little r, so we have homozygous dominant and homozygous recessive. We're going to also say that these flowers are part of the P generation. So we have our little key up here. So big R gives us a red flower. Little r gives us a white flower. And then big R gives us a pink flower. Did I say pink twice? Little r is white. Big R is pink. So it tells us our parents are big R, which is red, and little r, which is white. So we have a red flower and a white flower. So again, we're going to set up our Punnett square exactly the same as we have been setting it up. So we're going to take mom's genotype, and we're going to write it down the side of the Punnett square. We're going to take dad's genotype and write it across the top of the Punnett square. And then we're going to fill the Punnett square in. So in this box right here, we get a big R and a little r. In this box over here, we get the big R, we get the little r. This box again, we get the big R and we get the little r. And then this box right here, again, big R and little r. So we have 4 boxes or 100% as heterozygous. All 4 boxes have big R, little r. And if we come up to our key over here, big R, little r means that the F1 generation, so remember, this is the P generation, these parents right here, which means in here is the F1 generation. This means that 100% of the F1 generation is going to be pink, 4 out of 4 boxes. 0% of the F1 generation is going to be red. There are 0 boxes. And then again, 0 out of 4 or 0% will have white in the F1 generation. Okay, so incomplete dominance, we blended red and white together to make pink. Let's try this again. This time, it says, let's cross 2 flowers from the F1 generation. So if we come back over here, remember this is the F1 generation. So all 4 flowers here are heterozygous. All 4 flowers are pink. So we're really just taking for the F1 generation cross 2 of them. It doesn't matter. They're all the same. So this tells us that our parents will be heterozygous or pink. It doesn't matter because they're all the same. So same key, big R is red, little r is white, big R, little r is pink. Both of our parents are pink, and then we're going to set up our Punnett square to determine the genotype of the F2 generation. So we take mom, we write it down the side, we take dad, we write it across the top, and then we fill our Punnett square in. So big R for the first box, big R, little r for the second box, big R, little r for the third box, and then little r for the fourth box. So what we're seeing in this F2 generation is that 25% or 1 out of 4 offspring are going to be red. Right here, we have 2 big R's. 2 out of 4 or 50% right here are going to be pink because they are big R, little r. They are heterozygous. And then 1 out of 4, or 25%, are going to be white or homozygous recessive. So it's the same idea, it's just slightly different rules to determine what the phenotype is going to be. So again, let's continue with incomplete dominance, but use a human trait. So hair texture is actually an incompletely dominant trait for humans. So hair texture, we're talking about straight hair, curly hair, and wavy hair. So if we think about this, we have big C is going to be curly hair, little c is going to be straight hair, and then somewhere in the middle is wavy hair. So that's our heterozygous phenotype. Big C, little c is going to give us wavy hair. So it says here, hair texture follows an incomplete dominance pattern. Curly hair, big C, is incompletely dominant to straight hair, little c. That means that wavy hair is heterozygous, big C, little c. So that's what we did in our little key right here. Show a cross between a mom with curly hair and a dad with straight hair. So curly hair is big C, straight hair is little c, little c. And it says show a cross. So we're going to set up our Punnett square. So we're going to take mom's genotype and write it down the side. We're going to take dad's genotype, write it across the top. Then we're going to fill our Punnett square in. So big C, little c, big C, little c, big C, little c, and then big C, little c. So again, it's 100% big C, little c. It's 100% heterozygous. But remember, heterozygous in this case means wavy hair because incompletely dominant. So we have a third phenotype. So 0% is curly, 0% is straight. All of these offspring are wavy. So 100% wavy hair. Let's do this again. So again, it's going to say show a cross between two individuals from the F1 generation. I realize that because we're talking about humans, we are now inbreeding. This would be like a brother and a sister. So let's disregard that fact and just practice the Punnett square, okay? So if you remember from this Punnett square, all of the offspring were heterozygous, which means our two parents, because we're taking two from the F1 generation and making them the parents, they are both going to be heterozygous. They are both going to be wavy-haired individuals. So again, we write mom's genotype down the side. We write dad's genotype across the top. And then we fill in our boxes. So big C, big C. We have big C, little c. We have big C, little c. And then we have little c, little c. So in this case, we are seeing all three phenotypes. We have one box that is homozygous dominant. Those are our red C's, and that's going to be curly hair. So 1 out of 4 is 25% curly hair. We have two boxes that are heterozygous. That's the purple boxes. So big C, little c. That is wavy hair. So 2 out of 4 is 50% wavy hair. And then 25%, 1 out of 4 boxes, that's the blue box, are homozygous recessive. So that's little c with straight hair. All right, so let's talk about another inheritance pattern called codominance. So instead of the traits blending together in this example, both traits are going to show through for codominance. So if we think about like our flower example, instead of a red flower and a white flower producing a pink flower, a red flower and a white flower is going to produce a red and white flower. So we see this a lot in cow colors. So like the color of the fur of cows follows a codominant inheritance pattern. So you'll also notice that the alleles are written slightly differently for this version. We have a letter to represent the gene. So I'm gonna call this big R is gonna represent like fur color. And then these superscripts up here, the little letters up here, those are representing our different alleles. So let's actually look at this in an example. Like I said, we're going to be talking about the color of cow fur. So cows can be white. They can be red, which is like that like brownish color. So we have our white cow, we have our red cow, and then if you have a mix of both the white and the red, it is called roan. So this is your roan cow. So it says up here, cow fur color follows a codominant inheritance pattern. Cows can be red, which is written like this. So again, we're using the big R as our fur color. And then we are using this little r up here, the superscript, as the actual coloring. So we have fur color red, fur color red. So cows can be red, white, which is cow or fur color white, or cows can be roan, which is fur color red, fur color white. Roan cows are spotted red and white. So again, this is your roan color. So we have white, red, roan. And so I also have it in the key right here to show you what the different genotypes look like and what they mean for the phenotype. It says show a cross between a red cow and a white cow, and that this will be our P generation. So we take our genotype here, and this is where things get a little bit weird for some people. Mom's genotype does get written down the side again. So remember, this big R and this superscript R are one allele. So they don't separate from each other, they stay together, but we have to separate the big R's from each other. So we split it down the middle right here. So this is one possible gamete that mom makes. This is the other possible gamete that mom makes. We can do the same thing with dad. We write his genotype across the top. So again, the big R stays with its superscript, and we just separate the two R's from each other. So big R, little r stays together, and then big R, little r stays together. So again, this is fur color white, or fur color red, fur color red. So we fill it in very similar to how we filled in our other Punnett squares. We're still bringing our two alleles together. So fur color red comes over here, fur color white comes down here, and we get a genotype of fur color red, fur color white. We can do the same thing in this box. So we have our big R comes over, big R, little r comes down, and we get fur color red, fur color white. Same thing in this box. We bring this genotype or this gamete over here. This gamete comes down here. So we get fur color red, fur color white. And same thing in this last box here. We take this gamete, bring it over. This gamete, bring it down. Fur color red, fur color white. So when we're looking at all four of these boxes for the F1 generation, you're going to notice that they're all the same. So they're all fur color red, fur color white, which gives us our roan phenotype. So 4 out of 4 boxes, or 100% of this F1 generation, is going to be roan, which means 0% is going to be red and 0% is going to be white. In this next Punnett square, we're just going to cross 2 of the F1 generation. So all of the F1 generation is roan. So we're going to be crossing 2 roan cows. So again, we have our 2 roan cows. So here's roan cow 1, roan cow 2. We fill in our Punnett square. So mom's genotype gets written down the side. Dad's genotype gets written across the top. We fill in our Punnett squares. So we bring this over here. This gamete comes here for fur color red, fur color red. We take our gamete, bring it over, gamete, bring it down. So we get fur color red, fur color white. We take our gamete here, bring it over, gamete up here, bring it down for fur color red, fur color white. And then in the last box, we take our gamete, bring it over, gamete, bring it down for fur color white, fur color white. So when we look at the phenotypic ratios, we have one box right here that is fur color red, fur color red. So that tells us that our phenotype is going to be red. So 1 out of 4, or 25% of our F2 generation is going to be red. 50%, 2 out of 4, so 1, 2, is going to be roan. And then 25%, 1 out of 4, is going to be white. So that's going to be a codominant inheritance pattern. Let's look at an example in humans. And this one is a little bit more complicated, I should say. So we're going to be talking about blood types. Now, you might have heard this in terms of being able to give or accept blood. So if you want to donate your blood, you need to know your blood type. And if you need to be able to accept a blood donation, again, you need to know your blood type. So basically, this is telling us the different types of blood that are out there. This is a simplified version of it, but it still will work in the same way. So again, we're still talking about codominance, which means that type A blood and type B blood are codominant to each other. Type O blood is recessive to types A and B blood. So we can have 4 possible phenotypes. We can have type A blood, we can have type B blood, we can have type AB blood, and we can have type O. So like I said, type O is completely recessive to everything else. So we write that with 2 lowercase i's. I is just representing blood type. So if we have type A blood, we have blood type A, that would be homozygous for type A, or you can be heterozygous for type A. So you can be blood type A and then blood type O, so the recessive. Type B works the same way. You can be homozygous for type B blood, so you can be blood type B, or you can be heterozygous, so blood type B and then just blood type nothing. And then type AB would be your co-dominant, so you'd have blood type A, blood type B, and then like I said, blood type O is recessive, so just 2 lowercase i's. Now, what it's talking about when it's this A and B stuff, it's talking about proteins on the outside of your red blood cells. So if you have type A blood, you are going to have these A antigen proteins around the outside of your red blood cell. If you have type B blood, you're going to have these B antigen proteins around the outside of your red blood cell. And then if you're type AB, you're going to have some A proteins and some B proteins. And if you're type O, you have no proteins. And this comes into play when we're talking about the immune system and how your body is able to recognize self versus invader. But the idea here is that these proteins signal to your body that this cell belongs to the body and it's not a virus or bacteria that's trying to infect you. So the idea is if you are receiving blood from a different blood type, so let's say you're type A blood, all of your red blood cells have this protein around the outside, and you get type B blood, your body is going to see this as an invader and try to kill it. So if you're in need of a blood transfusion, you're already in some serious trouble. But then the blood that you're being given isn't even going to help you out because your body is going to recognize it as an invader instead of part of yourself. So that's why it's important to match blood types if you are donating your blood or you are receiving blood. If you are blood type O, you are known as a universal donor because anyone else can accept your blood because the body will recognize it as self. There's no proteins on it, so the body will just accept it as part of the body. If you have type AB blood, you can receive any type of blood because your body will always recognize it as itself. So again, if we come back to like if you're type B blood and you get type A, the body's going to be like, this A protein isn't a part of me, so I need to kill it. So that's why the type A blood won't work if you are type B blood. All right, so that's the little explanation of blood type. Let's actually do some Punnett squares with this. All right, so it just, again, says the same thing. Type A blood is codominant to type B blood, and then type O blood is recessive. So it says show a cross between a woman who is type O and a man who is type AB. So I have a little key up here telling us the different types of blood with the genotypes, and we have a little chart down here to help us out a little bit more. So when we have a woman who is type O blood, type O has to be little I, so I, Captain. Her genotype is II, and then if the person is type AB blood is IA, IB, so that's what we see right here. Again, we fill in our Punnett score the same way. So we take mom's genotype, II, and write it down the side. We take dad's genotype and write it across the top, so IA and IB, and then we fill in our Punnett square. So I comes over, IA comes down, so we get IAI. Again, IB comes down, I comes over, so IBI. IA comes down, I comes over, so we get IAI. And then IB comes down, I comes over, and we get IBI. So now when we are predicting the F1 generation phenotypes, we're looking at these 4 boxes right here. So 2 of these boxes are IAI, which tells us that 50%, 2 out of 4 boxes are type A blood. They are heterozygous for type A blood. And then 50%, 2 out of 4 boxes are going to be type B blood. They're heterozygous right here. So IBI. You're going to have 0% type AB and 0% type O. Let's try this again. So this time we're showing a cross between a woman who is heterozygous for type B and a man who is homozygous recessive. So heterozygous for type B. If we look over here, type B, hetero means different, so IBI is our first genotype, and homozygous recessive, that means 2 of the same, and the recessive allele, which is little I, so they're type O, so little I. So let's fill in our Punnett square. We take mom's genotype, write it down the side, IBI. Dad's genotype, write it across the top, I. We fill in our Punnett square, so IB, I, right there. So when we're looking at phenotypic ratios, we have 2 boxes, IBI, so that's going to be 50%, 2 out of 4, type B blood. And again, we have 2 boxes, 2 out of 4, or 50% type O blood. In this case, we have 0% type A and 0% type AB. So now that we better understand this idea of codominance and using the Punnett scores to help figure it out, we can play a really fun game of who is the dad. So if you've seen on like the Jerry Springer show or the Maury show, when someone comes on and they don't know who the father is and they do the DNA test or the blood test or whatever, this is how they determine who the father is on those shows. They use blood type to help you figure it out. So it tells us mom is type A blood, and we can figure this out through a blood test, which means she's type A. There's 2 possible genotypes. She can be homozygous dominant, or she can be heterozygous. And I'm showing that right here as well. Baby is type O blood. So baby right here, type O is II, what we see right here. Possible dad number 1 is type AB blood. So type AB is IAIB. That's what we're showing here with dad 1. And possible dad number 2 is type B blood. So he can be homozygous or he can be heterozygous. So homozygous type B would be IBIB. Heterozygous would be IBI. And we're showing that right here with dad number 2. So in order to figure out who the father is, we have to show a Punnett square. Now, we know that mom and baby are type A and type O. So mom is type A, baby is type O. Now, baby is type O, which means there's 1 genotype baby can be II. So baby had to get 1 I from mom and one I from dad. So based off of that information, we know that mom has to be heterozygous. She has to have this little I in her genotype to pass it on to baby. So mom is type A, she's IAI. She's type A for this IA, but she's heterozygous because baby is type O. If we look at dad number 1, he is type AB, so we're going to put his, baby is II, like I said. So we have to be able to get this genotype. Now, dad is IA, IB, and if we look at this Punnett square, there's no possible way that dad number 1 can donate a recessive I to baby to make it type O blood. So if we fill in the rest of it just for practice, this is what we would get. But down here, it would be IBI instead of II. So Dad number 1 can't be the dad. So let's try the same thing with dad number 2. So I'm looking at my options for dad number 2, and he's type B blood. We don't know if he's homozygous or heterozygous, but in order to make baby, he would have to be heterozygous because just like mom, he has to have a recessive I to donate to the baby. So as you can see here, here's mom, IAI. She's type A blood. She donates her recessive I to baby. Dad is type B blood. He has to be heterozygous because he has IB to make him type B, but he has to be able to donate his recessive I to baby. So this shows us, I'm just filling in the Punnett square now, but this shows us that dad number 2 has to be the father because he can donate his recessive I. Mom donates her recessive I, and that makes little baby II type O blood. So a little fun who's the father game. All right, we have one more inheritance pattern, and this is a sex-linked trait. So for sex-linked traits, this is a trait that is carried on the X chromosome, which means that this matters if you are male or female. So again, I'm talking biological male or biological female here. So if you are a biological female, and I'm going to refer to this as just female and male, just so I'm not with words. So if you are female, you have 2 X chromosomes. If you are male, you have 1 X and 1 Y chromosome, which means if this trait is carried on the X chromosome, it's more likely to show in males because they only get 1 copy of the X chromosome. Females get 2 copies of the X chromosome. So if a female gets a bad copy of a gene, she will still have 1 normal copy to cover it up. Males, on the other hand, if they get 1 bad copy of the gene, they don't have a second copy to cover it up. So an example of this is color blindness, specifically red-green color blindness. The idea here in this picture that it's showing you, if you are colorblind, if you look at these little circles, most of us will be able to see that there's a little like number in the middle of each circle. If you're colorblind, you won't be able to see the numbers. So if you're colorblind, this means you're missing receptors in your eyeballs that pick up red and green. They're called cone cells. So you are missing either red cone cells or green cone cells. And that means that your eyes are picking them up as the same color. So you see red and green as the same color. So that means if you're looking at these circles right here, you can't see that there's a 12, 8, 6, 29, 57, etc. This can be a big deal. It's not so much a big deal in like modern life because it's not. But back like in the wild specifically, so number 1, food and fruit. If it's ripe and ready to eat, it's more likely a reddish color rather than a green color. But also at the same time, things that are poisonous tend to be red as well. So if you're mixing up red and green, you won't know if a food is ready to eat, and you also won't know if something is poisonous. So that's why this can be a big deal. Some other examples of this are like, all right, so if you go out to like the Midwest of the United States, instead of their traffic lights being vertical with red, yellow, green, it's actually turned on its side. So it makes it a little bit harder to drive if you are colorblind, unless you already know if it's right or left for red or right or left for green. But like I said, not a huge deal. It's just you mix up red and green. It happens. So keep in mind, this is written similarly to codominance in that we have an X to represent the X chromosome, and then we have that little superscript letter to represent normal vision or colorblind vision. So in this case, the big R is representing normal vision, and the little r is representing colorblind vision. And we can't see the table, so there we go. All right, so now that we can better see our table, we can see female genotypes can be X big R, which would be normal vision, homozygous normal. We can have X big R, X little r, which again would be normal vision, but you're heterozygous, and you can be what we call a carrier, which just means you pass the trait, you can pass the trait on to your offspring. You can also be X little r, which would be homozygous recessive, and you would be red-green colorblind. If you're a male, like I said, males are XY. So a male with normal vision is going to be X big R Y, and a male with colorblind vision is going to be X little r Y. So that's what I mean when I say if a male gets the bad copy, in this case, the recessive copy for colorblindness, they're automatically colorblind. If the female gets one copy for colorblindness, she still has a chance of covering it up with a normal vision allele. All right, let's go back to present mode. All right, so let's practice this in a Punnett square. We are going to cross a normal vision heterozygous female with a colorblind male. So I still have our little table over here to help us out. But it says a heterozygous normal vision female. So heterozygous is one of each. So we have X big R, X little r for our female. And then we have a colorblind male. So he has to be X little r Y in order to be colorblind. We set the Punnett square up exactly the same. So we take mom's genotype and we write it down the side. So mom is X big R, X little r. Dad's genotype gets written across the top. So he's X little r Y. And then we fill it in the same way. So we bring this gamete over here, this gamete over here to get X big R, X little r. We bring this gamete over, this gamete down to get X big R Y. This gamete comes over, this gamete comes down for X little r, and then this gamete comes over, this gamete comes down to get X little r, Y. Now, in this case, the phenotypes get a little bit more difficult to figure out. It's a little bit more complicated. So not only do we have to pay attention to colorblind versus normal, but we also have to pay attention to male versus female. So in this box right here, XX tells us this is a female. This female has a big R, so she has normal vision. However, this female right here, because remember XX, this is X little r, so this female is colorblind. So we're going to have one box, 1 out of 4, 25% female normal vision. We're going to have 25%, 1 out of 4, female colorblind vision. Over here, XY, this is a male. He's X big R, so this is a male with normal vision. Down here, X little r, this is going to be a male with colorblind vision. So we have a 25% split across the board here. 25% of the offspring are going to be normal vision female. 25% are going to be a colorblind female. 25% are going to be a normal vision male. And 25% are going to be a colorblind male. All right, so it worked the same way. It's just a little bit more complicated in terms of what you're reading for the phenotype. Let's try another example. So in this case, we are crossing a colorblind female with a normal vision male. So colorblind female, here are our females, XX, colorblind is going to be X little r, that's what we have right here. Normal vision male, so male is going to be XY, and then normal vision is the big R, so X big R Y for our male. We take mom's genotype, we write it down the side. We take dad's genotype, we write it across the top. We fill in our Punnett square, same way we've been filling it in. Now let's take a second to figure out the the phenotypes. Okay, so again, this side is definitely our females. This side is definitely our males. We just have to determine if they are normal vision or colorblind vision. So this right here, X big R, X little r, is normal vision carrier. So normal vision, they can pass it on to their offspring. This female right here, same thing. So 2 out of 4, 50% chance of a female with normal vision, 0% chance of a female with colorblind vision. Over here, we have 2 males. So X little r Y is a colorblind male right here, red-green colorblind. We have 2 boxes of those. So again, 50%. 2 out of 4 are going to be colorblind males, and there's a 0% chance of producing a normal male. So in this case, either these parents are going to have a normal vision female or they're going to have a colorblind male. Those are their only two options. All right, and so this is what I already talked to, but because color blindness is a recessive trait that is sex-linked, it's carried on the X chromosome. Females, like we saw, get 2 copies of the traits on the X chromosome, so even if they have a colorblind copy of the allele, they can still have a normal vision copy of the allele, and it cancels it out. So they will have normal vision. Males, on the other hand, they only get the 1 copy. So if they get the colorblind copy, they are automatically colorblind. So that's why it is more common to see these sex-linked traits appear in males than in females. All right, so I'm going to stop here. Hopefully you were able to pause the video and do some practice Punnett squares. You can always go back and watch the video for the different segments while you practice your different types of Punnett squares. But at this point, that's what you should be practicing. Also, make sure you are continuing to fill in your vocab notebook if you haven't finished that as well, just the different types of inheritance patterns.